Assalamu alaikum. So in this lesson, we are doing objective still 16.2, the roles of genes in determining the phenotype. But we will be covering objectives E, F, and G, which say that explain the gene mutation occurs by substitution, deletion, insertion of base pairs in DNA, and outline how such mutations may affect the phenotype. That outline the effects of mutant alleles on the phenotype in the following human conditions: albinism, sickle cell anemia, hemophilia, and Huntington's disease. And finally, we'll talk about the relationship between genes, enzymes, and phenotype with respect to the gene for tyrosinase that is involved with the production of melanin. So gene mutation is actually a change in the sequence of base pairs in the DNA molecule that may result in an altered polypeptide. So we know the DNA molecule is made up of base pairs which exist as codon or triplets. So there can be sudden changes in the sequences of those codons, right? So if there is a sudden change, which can be heritable or non-inheritable, this would lead to a genetic mutation, right? So the agents which are causing mutations are known as mutagens. So these can be physical, like ultraviolet rays, or they can be chemical, like food adulterants, or biological, like certain natural food items. So remember, it is a change in the sequence of base pairs, right? So this is what mutation is. And it will be the base pairs which are there in the DNA molecule, and it may result in an altered polypeptide. And so because of the sequencing getting changed, we can have a different protein, right? So mutations actually, they occur continuously. But what is happening is that as this DNA-based sequence, it determines the sequence of amino acids that make up the protein. Mutations in the gene can sometimes lead to a change in the polypeptide that the gene codes for. But most of the mutations would not alter the polypeptide at all, or even if they are actually altering it, it would be so slight so that the structure of the protein would not change, right? Or the function would not change, right? Because the genetic code is degenerate. Now, as we're using the term degenerate, so understand it from this DNA code here. So here you're looking at, so here we are looking at a codon, right? So it's thymine, 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 right? And then you can look at thymine, thymine, cytosine, thymine, thymine, adenine, thymine, thymine, guanine. So here you can see the first two, like the nitrogen base are the same, but the third one has changed. So if TTT was coding for the amino acid phenylalanine, so even if this T gets substituted by a C, it is still coding for the same amino acid, phenylalanine, right? Then if you look here in the third one, thymine, thymine, adenine. So here if adenine gets substituted by a G or guanine, so still it is coding for the same amino acid, leucine. So this is what is meant by the term that the genetic code is degenerate. And the advantage for this is that although there is a mutation in the codon, if the mutation changes the codon to another codon, which codes for the same amino acid, this prevents against the mutation affecting the functionality of the protein. So you will still have the same amino acid and it will code for the same protein, right? So mutations in the DNA-based sequence can occur due to three different ways. So one of them is known as insertion, second is deletion, and third is substitution, right? So these can occur at the basis of any change in the sequencing of the nucleotide base, right? And we'll talk about these in detail one by one. So here you can take a look again. For example, at this part. For example, here and even in this case, all of them are coding for valine, right? So this is our DNA code, and this is an RNA code here that you're looking at. So here we have uracil, but still different codons, right? They're coding for the same amino acid. So you can see here, right? So that's why mutations, they happen, but all of them would not be harmful. All right then. So let's talk about the gene mutations. So we will be discussing three: substitution, deletion, and insertion. So let's use this. So substitution, as the name itself implies, it's actually one base substitutes another base. It takes the place of another base, right? So when one base substitutes another base, which misleads the base pairing. For example, again, if you're reading it as a triplet, like ATG, so see, this G has been substituted by T in this case. So now it is ATTC, right? So this is actually only one base substitutes another base. So this is what is known as substitution, right? Now, the second type of gene mutation is deletion. So again, the name itself is self-explanatory. It means removal of one or more bases, right? So substitution की जब हम बात करते हैं, we're talking about कि there's एक है सिर्फ, ठीक है, that gets substituted, right? But deletion में क्या हो रहा है? Removal of one or more bases. ठीक है, it can be one, it can be more, which will also alter the base pairing. For example, in this case, again, if you're reading it as 3ATG, right? And then we have GCT and then AAC. So if like one of this whole, like one base or maybe more bases are deleting, so see what happens. Like here in this case, it seems like this entire GCT has been deleted. See, this entire GCT, it's not here. We have ATG and we have AAC, right? So 3 bases have been deleted. So this might be a coding for a particular amino acid, and now because it's not there anymore, so it will eventually code for a different polypeptide. The third one is insertion. And insertion simply means addition, but here also is addition of one or more bases in the sequence of bases. For example, here in this case, if it is ATG and AAC, so now look, GCT has been added, right, or inserted. So 3 bases GCT are added. So it means definitely now maybe this amino acid was not supposed to be there, but it will be there now. But this also leads to a wrong base pairing. So the sequence of bases in DNA codes the sequence of amino acids in a protein. Please remember that. So any mutation in base or nucleotide sequence will alter the combination and sequence of amino acids. What is happening because of that? The primary structure of the protein will actually change. The primary structure that was intended to be formed, it will not change. It will be different from the original one. So the change in primary structure of the protein will ultimately change the 3D structure, right? Which cannot bind to active sites of the respective enzymes, or they may bind to another enzyme. This will result in wrong cascade of reactions in the cell and eventually will lead to diseases, right? So this is the impact then different types of gene mutations can have. So we have talked about substitution, deletion, and the third one that we discussed was insertion. All right, so here actually we are looking at one example from an insertion mutation. So here they have shown you an original D gene. So see, we read them as triplets. So it's TAT is coding for amino acid tyrosine, AGT is coding for serine, and then CTT is coding for leucine. But here in this case, you see here, insertion means addition of one or more base pair. So what has happened here is AG, there was a T that was supposed to come, but instead of this T, we have a G, right? And this T has shifted, right? So there is a frame shift as well. Since it like if it read it as a triplet code, so TAT, but AGG will now code for a different amino acid, which is arginine. And because of that, what happens? See, the third one in the original one was a CTT. It was leucine. But what happens now? Since D, like in the space of T, like a new one has been added, which is G. So T moves to this side now. And now this will be read as TCT. So this will code for serine. See? So this sequence will also change eventually, right? So the ones before that, like TAT will be normal, right? But this one here, where the insertion has taken or gene mutation has taken place, this one and the next one in the series after that will all be changed, will all be altered. Okay, so let's talk about substitution of nucleotides, right? For one type of genetic mutation, let's talk about that in detail now. So substitution is a mutation that occurs when a base in the DNA sequence is randomly swapped for a different base, right? And generally there will be only substitution में सिर्फ एक nitrogenous base है, right, that gets substituted. So unlike an insertion or deletion mutation, substitution mutation will only change the amino acid for the triplet, right? So it means that जो भी change है, it is there at the group of free bases in which the mutation occurs. ठीक है, but it will not have a knock-on effect. So substitution mutations can take three forms. And like as this term is written here, knock-on effect. So for example, here we were discussing insertion. So insertion में what is happening, this T, it was not removed at all. It was still there. ठीक है, but वो move करके यहाँ पर आ गया. ठीक है, ऐसा नहीं कि वो खत्म हो गया. ठीक है, so that's still there. ठीक है, substitution में what happens कि अगर ये T है, इसकी जगह G लग गया. ठीक है, कोई नहीं मतलब अलग से ऐसा नहीं हुआ कि it has been added. It has simply been substituted. Right? Or in fact, you can see here also. Like, so the first one, tyrosine, then serine. So see what has happened is in place of T, it has been replaced by adenine. Right? It is coding for a different amino acid, arginine, but nothing has happened here. This is leucine, it stays the same. Right? So it is only substituted. ठीक है? एक की जगह दूसरा लग गया. So that's the reason we say it does not have a knock-on effect. Knock-on effect या frame shift ये चीजें इसमें नहीं हो रही है. So substitution mutations can take three forms. One of them is silent mutation, then we have missense mutation, and then nonsense mutations. Okay then, what is happening in this silent mutation? Mutation does not alter the amino acid sequence of the polypeptide. This is because certain codons may code for the same amino acid as the genetic code is degenerate. And this we have already discussed in the previous slide. If I go back and show you, for example, here in this case, you see that various, like we have different ways. The first two are the same, right? So there's a change in the sequencing of the third base, but still they're coding for the same amino acid. Okay? So this type of mutation is known as silent mutation, right? And this is a substitution mutation. Okay, it's a silent mutation. So mutation would not alter the amino acid sequence of the polypeptide. Achha, what is happening in missense mutations? Mutation alters a single amino acid, right, in the polypeptide chain, okay, but it can cause a disease, right? So sickle cell anemia is an example of a disease caused by a single substitution mutation changing amino acid in the sequence, right? So in the sequence, one amino acid has been changed, right? So that is known as missense. Yeah, but it is causing a disease. Now, nonsense mutation, what is happening in this case? The mutation creates a premature stop codon. Okay, because like as these amino acids are made, so there is always initiation once the process starts, right? And then we have termination as well, right? So this termination is actually being like done or controlled rather by the stop codon. Okay, so जब वहाँ पे जब stop codon लग जाएगा तो the process will terminate और amino acid का sequence जो बनना था that would stop, right? So what is happening in this case, the mutation creates a premature stop codon which signals for the cell to stop translation of messenger RNA molecule into an amino acid sequence, causing the polypeptide chain produced to be incomplete and therefore affecting the final protein structure and function. And nonsense mutation ki example that we use is cystic fibrosis. So it is an example of a disease caused by a nonsense mutation, although this is not always the only cause, right? But here in this case, what is happening, a stop codon will attach there. So see, I'll just show you. For example, here, if you're looking at this RNA codon here, see, UAA, UAG, they act as a stop codons, right? So for example, some other nitrogen base sequence was going to attach there, but instead these actually go and they attach there. So this will be a stop codon. This will stop the transcription, right? So this is UAA, UAG, and UGA. They act as stop codons, right? Similarly, if you look at this DNA code here, so TAA, TAG, and TGA. So they act as a stop codons, right? So this will actually cause the premature, actually, how do I put it? So the protein production or protein formation would be incomplete, right? So you would not have the final protein structure that was required. If the protein primary structure is not there, right, it's incomplete, so it will not be able to perform its function, right? So these are three types of mutations, and basically substitution mutations. So it can be silent mutation, right? When the amino acid sequence, even if there's a mutation, it is not causing any change in the amino acid sequence because it is coding for the same amino acid, right? The genetic code is degenerated. Miss sense में क्या हो रहा है? It is altering a single amino acid alter भी होता है, so it is causing a disease, right? So sickle cell anemia is one example. It's coding for a different amino acid, right? Or eventually change हो जाएगा सब. Then nonsense mutation में क्या हो रहा है? The mutation creates a premature stop codon, right? And you will have an incomplete protein. If there is an incomplete protein, it will not be able to perform its function, right? So again, the same diagram as we've already discussed. So this is substitution, right? So this is T has been substituted by A. So we have a different amino acid in the sequence now. So the initial sequence was tyrosine, serine, leucine. Now it is tyrosine, arginine, leucine, right? So it has coded for a different amino acid, right? So in this case, this mutation can cause a disease as well, right? But for instance, if it was like coding for the same, there was a difference here in the sequencing, but even if it was coding for the same amino acid, okay, so here as such ज़्यादा problem नहीं होता, and it would have been a silent mutation. Okay, so we already talked about silent mutations. So it's a change in the nucleotide sequence that results in the same amino acid sequence. So this is possible because some amino acids can be coded for by up to 4 different triplet codon sequences, and we have just seen that. Again, I'll just show you. So see, for example, these. Even there is substitution, but all these 4 are coding for the same protein, same amino acid, sorry. Here you can see as well. See, even here you can see. So all of these are coding for valine, right? So silent mutations are often a change in the second or third base in the codon, right? So it's not the first one. So it could be second or third one. The ones that we have seen, so there was generally the change was in the third base, right? For example, valine is coded for by 4 different triplet codon sequence, and we just saw that as well. But see, GUU, GUC, GUA, and GUG. Even in this case, you see first and second are the same. जो change आया है, that is there in the last one, the third one. So it had to be U. Here it has been substituted by a C, substituted by an A, substituted by a G, right? So therefore, as long as the first 2 nucleotides in the codon are guanine and uracil, the amino acid valine will be inserted into the polypeptide, right? So this is about the silent mutation. Okay, now let's talk about the effects of these mutations. So we can have neutral mutations, right? We can have beneficial mutations and harmful mutations, right? So let's talk about the neutral mutations. Neutral mutation means that the protein which is produced by this type of mutation does not harm the organism or do not cause any disease. Because even if there's a mutation, right, the sequencing of base pair changes, right? It codes for a different amino acid, right? So the protein that is produced with this type of mutation, if it does not cause any harm to the organism, it is not causing any disease, right? So we call it as a neutral mutation. So there are 3 ways in which a mutation or its side effects can be neutral, right? So various triplets of bases or codons can code for the same amino acid. So अभी हमने ये substitution के अंदर silent mutation में देखा. Therefore, if mutation in base forms such a codon which codes for the same amino acid, like that of an unmutated codon, then it is a neutral mutation. For example, TTA and TTG, they're coding for leucine. And we have already seen that in that DNA code diagram. If TTA mutates to TTG, there is no effect on the formation of amino acid and its function, right? Different amino acid is produced as a result of mutation, but the amino acids produced are chemically similar, right? ठीक है, mutation हुआ है, sequencing में change हुआ है, we are getting a different amino acid, but even then, amino acid जो बना भी है, they're chemically similar, right? For example, AGG is mutated to lysine. AGG is coding for arginine. Arginine is amino acid. तो अगर substitution हुआ है, तो G की जगह यहाँ पे A लग गया है. So it is had mutated to lysine. ठीक है? So it is chemically similar, right? As such इनके function में बहुत बड़ा कोई effect नहीं आएगा. Then mutation results in such an amino acid which does not have any role in the protein function, as it is far away from the active site of the enzyme. ठीक है, mutation हो भी गया है, different amino acid बन भी गया है, but it is not necessary that it will have a role in protein function, right? If it does not have a role in protein function, ठीक है, as it is far away from the active site of the enzyme, because enzymes जो है, ठीक है, उनके एक्टिव साइट के ऊपर ही सबस्ट्रेट लगेगा, एंजाइम सबस्ट्रेट कॉम्प्लेक्स बनेगा. अगर तो उस म्यूटेशन से उस जगह पर कोई फर्क पड़ ही नहीं रहा है, ठीक है, सो अगेन दैट वुड बी अ न्यूट्रल म्यूटेशन, राइट? सो न्यूट्रल म्यूटेशंस क्या है? These are the types when the protein produced does not harm the organism or do not cause any disease. Second type of mutation is beneficial mutation. So beneficial definitely means it is advantageous, right? It has an advantageous effect on the survival of an organism. So for example, in certain bacteria, the enzymes can break down the antibiotics, right? This means that they have developed resistance against the antibiotics to mutation. Yeah, because antibiotics का क्या काम होता है? They kill the bacteria, but these bacterial enzymes, ठीक है, they have mutated, क्योंकि definitely उनके base sequence में mutation हुआ है. They're coding for a different amino acid, right? So definitely they're coding for different proteins or enzymes. So they can break down the antibiotics, and they have developed resistance against the antibiotics to mutation. So अब वही antibiotic उनके ऊपर अब काम नहीं कर रहा है, ठीक है. So breakdown of antibiotics, अगर if you look at it from the bacterial perspective, bacteria के perspective से देखो, so it is beneficial for them, right? Third type of effect जो है mutation का, that is harmful mutation, ठीक है. Harmful definitely इसमें तो disadvantage है, ठीक है, and disadvantage है basically in the survival of organism. So it decreases the chances of survival. So harmful mutation generally होती हैं deletion की वजह से, right? So removal or deletion of codons that codes for CFTR gene. So there's this particular gene there. Okay? It can cause cystic fibrosis, leading to the formation of thick mucus, which can lead to the persistent lung infections and complications in other organs. Right? So again, if this gene is not there, right, it will lead to thick mucus production. ठीक है, और thick mucus बन रहा है, तो it is blocking the lungs, ठीक है, causing lung infections, and it is causing complication other organs as well. ठीक है, तो ये एक harmful किस्म की mutation है. Clear? All right. So the 4 diseases caused by genetic mutation that we have to talk about, as per our syllabus, is hemophilia, albinism, sickle cell anemia, and Huntington's disease. So let's talk about what happens in hemophilia. So hemophilia is actually caused by a mutation in the gene F8, which codes for the factor VIII protein. So factor VIII protein actually is a coagulating agent. So it actually plays an essential role in blood clotting. And what happens in hemophilia is that we have abnormal alleles of the F8 gene that can then result in production of abnormal forms of factor VIII, because the gene F8 is coding for factor VIII protein. So if there are abnormal alleles of the same gene, so we can have production of abnormal forms of factor VIII. There can be less production of the normal factor VIII, or then there can be no production of factor VIII at all, right? So if there is a lack of normal factor VIII, it would prevent normal blood clotting, and this will lead to bleeding, and the disease that will be a condition that will be that is caused is known as hemophilia. So hemophilia is actually a sex-linked disease because this F8 gene is located on the X chromosome. So hemophilia, yes, we say it's a sex-linked disease. And the thing is that if males have an abnormal allele, they will have the condition. So they will be hemophilic if the male has that allele, right? Because he has X and Y chromosome. So if he has the abnormal allele from the X chromosome, he will have the condition, right? Because they definitely only get one copy of the gene. Females can be heterozygous because they can have a normal allele and one abnormal for the factor VIII, and they might not suffer from the condition, but definitely they will be a carrier, so they will act as vectors, right? So when we look at the parental phenotypes, for example, let's say we have a normal male. So if you look at the phenotype, it's X, H, capital H for the normal and Y. And for the carrier woman, so you can see she has one normal allele and one abnormal, one hemophilic. So capital H and small h, right? So if you look at the gametes, and then as we get this Punnett square done, so we see there can be one normal female, there's a normal male, right? There's a carrier female and one hemophilic male, right? So these are the gametes. If there's a cross between a normal male and a carrier woman, so there's a chances of male. So there are chances of a hemophilic male only when the mother is a carrier, right? So normal male, right, and carrier woman. So actually, it is because of the carrier woman, right, or mother, right, which is actually passing this abnormal allele to the male offspring, and we are having this hemophilic male. So if you look at the diagrams here, so they have shown you for the healthy blood vessel. So there's a normal blood vessel, and if let's say hemorrhage occurs, so because of the clotting factor, it will eventually lead to clotting and no more bleeding happens. But in case of hemophilic, normal blood vessel is there, hemorrhage occurs, but the ability of clotting, your ability to clot is actually minimized or is not there, right? So this is what happens in hemophilia.
Hemophilia is a X-linked genetic disorder in which blood doesn't clot and bleed longer. Hemophilia is caused by a dysfunctional or absent blood clotting protein. Without this protein, stable clots do not form quickly over wounds. Weak clots are formed, but they are easily dislodged. To understand this, first we need to understand details of clotting. The clotting process starts after the wall of the blood vessel is breached. The vessel constricts to reduce blood flow, and the platelets assemble at the damaged site. Though the platelets stop the bleeding at this stage, they are easily dislodged, and the bleeding can resume. As the platelets arrive, molecules released from the damaged vessel activate clotting factor 12. Factor 12 then activates another clotting protein, the factor 11. By closely fitting into the it-like lock key mechanism. Once factor 11 is activated, it activates the next molecule, factor 9, with the same lock and key mechanism. This initiates a cascade of reactions that ends when the last molecule, thrombin, is activated. Activated thrombin then snips small pieces off another protein called fibrinogen. When lots of fibrinogen is cut, the prune molecules cover the platelets and stabilize the clot. To form a stable clot, all the molecules in the cascade must be present and properly shaped. There are two main types of hemophilia, hemophilia A and hemophilia B, which are further divided into moderate to severe types. Hemophilia A. Mild or moderate hemophilia A. In a mild or moderate hemophilia A, factor 8 is present but has a slightly dysfunctional shape that sometimes doesn't fit into the next molecule. Because the mutated factor 8 succeeds in activating factor 10, so the people with mild or moderate hemophilia A do not bleed as long when injured and only rarely, or never, bleed spontaneously. People with mild hemophilia A have mutated factor 8 proteins. This is due to a small mutation in their factor 8 gene, located on X chromosome. The gene's code carries the instructions for building the protein, so a minor change in the instructions causes a minor change in the shape of the protein. Severe hemophilia A. In people with severe hemophilia A, factor 8 is usually absent, and the clotting cascade comes to a complete halt. Factor activity is less than 1% of normal. People with severe hemophilia A bleed spontaneously and can bleed for days after minor injuries. In people with severe hemophilia A, factor 8 is not present, because their gene contains a much bigger mutation, like an inversion, that completely garbles the instruction for protein. No factor 8 can be produced from garbled gene. Hemophilia B. Mild or moderate hemophilia B. If factor 8 is normal, but the person still has hemophilia, the change in factor 9 is usually the culprit. The disorder is called hemophilia B. In a mild or moderate hemophilia B, a small mutation in the gene leads to a slightly dysfunctional factor 9. The protein works occasionally to activate thrombin, and stable clots are eventually formed. Severe hemophilia B. When hemophilia B is severe, a more critical mutation in the gene completely misshapes the factor IX protein. The protein either cannot activate it or cannot activate next molecule in the cascade. Before effective treatments were developed in late 60s, people with severe hemophilia died young from bleeding. With effective treatment, life expectancy is nearly normal, but repeated bleeding episodes can cause disabling arthritis in the joints. Damage to the joints, mainly knees, elbows and ankles, starts when the synovium, which is a thin lining inside the joint capsule, thickens to absorb blood, lost from the vessels. As the synovium thickens, it also acquires more blood vessels that make the joint prone to further injury. Another injury means more blood to absorb, and the synovium thickens again. With frequent re-injuries, the synovium never shrinks and remains swollen. Enzymes from the swollen synovium eats away at the cartilage that cushions the ends of the bones in the joint. The longer the synovium stays swollen, the more damage occurs. Eventually the cartilage is eaten away and the bone begins to grind against bone, causing pain and reducing the joint's range of motion.
So in the video that you watched, there was detailed information about factor IX as well. But right, as per your syllabus specification, you may stick to only information regarding factor VIII, right? And we are also not differentiating into hemophilia A and B as per syllabus specification. We are only talking about hemophilia in general. Okay, so stick to what is in there in your syllabus actually. All right, then the second disease that is caused by the genetic mutation that we have to talk about is albinism. And here there is a mutation in TYR gene, right? So humans who have albinism, they lack the pigment melanin in their skin, hair, and eyes, right? So the pigment is not there. So because of that pigment not being there, these people have pale skin, their hair is pale, and they have pale blue or pink irises in the eyes, right? So, but what is happening is that there is actually some defect in the metabolic pathway for the production of melanin. The end product is melanin, right? So the defect is there in that the enzyme tyrosinase is actually the enzyme tyrosinase actually is missing, okay? Or actually like there is some deformation there. So the amino acid, it starts with the amino acid tyrosine, right? So this is the starting point. And this tyrosine has to convert into DOPA. DOPA has to convert into dopaquinone and finally it turns into melanin. So melanin is the end product. So what is happening is there the enzyme tyrosinase, which actually catalyzes the reaction of conversion of tyrosine to DOPA and then from DOPA to dopaquinone is not there, right? So this enzyme is missing, tyrosinase. So DOPA has to convert to dopaquinone, okay, again by the same enzyme. So the same enzyme that is catalyzing the reactions. So its tyrosinase is required to catalyze the reaction from tyrosine to DOPA, and that is also required for converting DOPA to dopaquinone, right? But it's not there. So the gene that is located on chromosome 11, TYR, right? This gene is located on chromosome 11, and this codes for the enzyme. The thing is that this gene, if there's a recessive allele for the gene TYR, that causes a lack of enzyme tyrosinase or the presence of inactive tyrosinase, right? So it actually, because of the recessive allele. So the recessive allele for the gene TYR, it causes a lack of enzyme or presence of an inactive enzyme. So if the enzyme is inactive, also then it is not able to perform its function. Definitely is not able to catalyze the reactions there. So without this enzyme, tyrosine cannot be converted to melanin, right? So if the pigment is not being formed, the condition that will lead, that it will be leading to is albinism. So the gene is TYR gene and it's recessive allele that is causing the lack of enzyme tyrosinase. The third disease that is caused by a genetic mutation that we have to talk about is sickle cell anemia. And the gene that is involved is HBB gene, right? So sickle cell anemia is a condition that actually causes individuals to have frequent infections. They have severe episodes of pain as well as anemia, right? And the thing is that people with sickle cell anemia have an abnormal hemoglobin, right? So the hemoglobin, okay, is abnormal in the red blood cells, and the abnormality is actually that there is a defect in beta globin. So beta globin is a polypeptide which is found in the hemoglobin, and this beta globin actually codes, it is coded for by the gene HBB, right? And this gene HBB is also found in chromosome 11, right? So this gene HBB is coding for this beta globin polypeptide. So if there is an abnormal allele for this gene HBB, what will it do? It will produce a slightly different amino acid sequence to the normal allele, right? So because of that, there will be change in the DNA base sequence, okay? And it will result in an amino acid substitution. So sickle cell anemia actually is an example of a substitution mutation, right? And what happens is that the base sequence CTT, right? So it is replaced by CAT. So CTT is coding for the amino acid glutamic acid, right? And CAT is coding for valine. So there is only substitution in sequencing of one amino acid, right? So this results in an abnormal beta globin polypeptide, right? So instead of the normal beta globin, so there's an abnormal beta globin now. So abnormal beta globin and hemoglobin affects the structure and shape of the red blood cells, right? So, and the shape actually becomes such that these cells are pulled into half moon shape. They're unable to transport oxygen around the body because normal red blood cells, like they can carry oxygen, okay, and then it can be transferred to different parts of the body. But these shapes, which have become sickle shape, or they have this half moon shape, they're unable to transport oxygen around the body, right? And they also stick to each other and then clump together, blocking the capillaries. And this is what can actually lead to the pain as well. So a person who is homozygous, right, and has two abnormal alleles for HBB gene will produce only sickle cell hemoglobin, right? So they will have only sickle cell hemoglobin if the person is homozygous, right, for sickle cell anemia. So these people will have the disease. They will have sickle cell anemia and they will suffer from the associated symptoms, right? But a person who is heterozygous, who is having a normal allele and an abnormal allele for the HBB, that person will produce some normal hemoglobin and some sickle cell hemoglobin. So these people will be carriers, right? They will have no symptoms at all, right? Unless they go to a high altitude where oxygen concentration is less, or they have this vigorous exercise where their oxygen concentration becomes less, then they will start having the symptoms. Otherwise, they will stay normal. These alleles actually they are co-dominant, right? So we can have hemoglobin A. So a person who is having homozygous normal, right? The person could be homozygous and containing that normal allele, both for sickle cell. So this person will have the sickle cell disease, right? This is the normal individual. And then we have this person who is a carrier, right? So this person is having the normal allele as well as the sickle cell. So this person will actually have the sickle cell trait, right? And this person will have the sickle cell anemia. This person will have the disease. This is a normal individual.
Sickle cell anemia is an inherited disorder that affects the red blood cells. Because it is inherited, it means that it can be passed from parent to child. So what is it? Red blood cells contain a red protein called hemoglobin, which carries oxygen around the body. Normally, red blood cells are round and flexible, which means they can bend to move easily through the narrow blood vessels. People with sickle cell anemia have a change or mutation in the hemoglobin gene. This faulty version of the gene causes the hemoglobin to form strands in the red blood cells, which makes them inflexible and gives them a crescent or a sickle shape. This is where the name of the disorder comes from. A sickle is a tool used to cut long grass. These unusually shaped red blood vessels can cause blockages in sections of the blood vessels. Red blood cells cannot pass through the blockages to reach the cells in tissues and organs, so the cells cannot get oxygen and glucose for respiration. When this happens, it causes pain called a sickle cell crisis. The symptoms of a crisis can be severe and last from a few minutes to several weeks. It can cause complications, which can be life-threatening. For example, if a blockage occurs in the blood vessels in the brain, then brain cells can die, which causes a stroke. So, how is sickle cell anemia inherited? Well, to have the disorder, you have to have 2 copies of the faulty allele, which is a recessive allele. People who have 1 copy of the recessive allele are carriers. The red blood cells of carriers have 50% abnormal hemoglobin and are slightly more fragile than normal. If these 2 sickle cell carrier parents have offspring, they can have children with the disorder, children who are also carriers, or children who are completely unaffected, which we can show in a Punnett square. There are about 4.4 million people in the world who have sickle cell anemia, while an additional 43 million are carriers. About 80% of people with sickle cell anemia live in Central Africa. It also occurs relatively frequently in parts of India, the Middle East, and among people of African origin living in other parts of the world. These regions are also most affected by the disease malaria. This is not a coincidence. Having the sickle cell allele actually protects you against malaria. This means you are more likely to survive childhood and pass on the sickle cell allele to your children. This also means that the numbers suffering from sickle cell anemia are also higher in malaria regions, because if 2 carriers have children, they have an increased risk of having children with the disorder. But how does carrying the sickle cell allele help protect against malaria? The malarial parasite is transmitted by female mosquitoes, which spend part of its life cycle inside red blood cells. When these parasites enter the fragile red blood cells of carriers, the cells often burst before the parasite has time to develop. This means the parasite dies, and the life cycle is broken. So, in this video, you have learnt what sickle cell anemia is, how it is inherited, and why it is more common in some areas of the world than others.
So the 4th disease caused by genetic mutation that we have to talk about in our syllabus is Huntington's disease, right? And it is caused by a mutation in HTT gene. So you're required to actually learn the names, right? And how the disease actually occurs and where the mutation happens. So this HTT gene and Huntington's disease, right? So this disease actually is genetic, and it actually develops as a person ages, right? So usually a person with the disease will not show any symptoms until they are 30 years of age, right? So though it is a genetic disease, but it shows its effects when a person, like he ages, and it is like he becomes more than 30 years of age. So the person who has the condition then experiences neurological degeneration. They lose their ability to walk, talk, and think, right? And this disease ultimately becomes very fatal. Definitely, it's fatal. So it has been found that individuals with Huntington's disease have abnormal alleles of HTT gene, right? The HTT gene code for protein Huntington. Okay, so this is the protein that this gene HTT is coding for, and it is involved in the neuronal development. So people who have a large number of repeated CAG triplets, right? And when we're talking about large number, we're talking about more than 40, right? So they have these more than 40 repeated CAG triplets. They are present in nucleotide sequence of the HTT gene. They suffer from the disease, right? So they have more than 40 or repeated CAG triplets, right? So the abnormal allele is dominant over the normal allele, right? If an individual has one abnormal allele present, they will suffer from the disease. So it is not that कि जी homozygous होगा तो सिर्फ उसी को हो रहा है, ठीक है? So even heterozygous है बंदा, so he is also suffering from the disease, right? Because it's dominant over the normal allele. So here there's a chart that you can use actually as a recap, right? As you're learning about these diseases. So first up, okay, about the disease albinism. The gene involved is TYR gene, right? It is actually coding for the enzyme tyrosinase, right? And that this is the enzyme, and eventually, if the enzyme is not there or it is inactive, we will not get the product which is melanin. Melanin will not be produced. So there is there will be a defect in the metabolic pathway of getting melanin. So the pigment will not produce. Okay, and the person will have albinism. So these people they are homozygous for abnormal allele, right? Now coming to sickle cell disease, basically, right? So the key involvement is there for hemoglobin. So hemoglobin abnormality is there at the beta globin polypeptide, right? So the gene that is coding for the beta polypeptide is HBB gene, okay? And right, if there's an abnormality there, so it is coding for a different base sequence, right? So instead of glutamic acid, valine is produced. And if the person is heterozygous, he will be a carrier, right? And if the person is homozygous for abnormal allele, the person will have the disease. He will have sickle cell anemia, right? Then coming to hemophilia, right? So hemophilia, actually, the key molecules are involved in factor 8. Factor 8 is a coagulating factor. It's a protein, right? And the gene that is coding for factor 8 protein is F8. So if there is an abnormality in F8 gene, which is actually sex-linked, right? So if it is homozygous for abnormal allele, the person will have hemophilia. In this case, actually, the females, like the females which have this hemophilia, they would not exist at all because they would die, like in the fetal stage or as soon as they they are born, because this is lethal, right? So a female having two abnormal alleles cannot survive at all, right? Because the blood cannot clot at all, right? So and if it's a male, right? So male will have the disease, right? And heterozygous female will be a carrier, so she can survive and then she can pass on the disease to her son, right? Then coming to Huntington's disease, in Huntington's disease, the protein that is involved is Huntington, and the gene that is coding for this protein is HTT gene. And in this case, it is actually the homozygous. If the abnormal allele is homozygous, the person will definitely have the disease. But in this case, heterozygous will also cause the disease because it is dominant, right? So if the person is heterozygous, he's having one normal and one abnormal allele, even then the person will have Huntington's disease. So this disease usually manifests after 30 years of age, right? And so these are the 4 genetic mutations. Okay, the diseases that we have to learn about. You need to know the genes involved. You need to know the proteins they're coding for, right? And you need to know the genotypes, right? Okay, so in your exams, you may be asked to predict the inheritance of a disease, like the ones that we have just talked about: albinism, sickle cell anemia, Huntington's disease, and albinism. So an example question could be that Max and Jane are trying for a baby, but they're concerned about the possibility of the child having hemophilia. Neither Max nor Jane, they have hemophilia themselves, but Jane's father had the condition, right? He had the condition, right? So what are the chances that the child could have hemophilia, right? So for questions like this, it's important that you gather on information whether the abnormal allele that caused the disease, is it dominant or is it recessive, right? If there is any sex linkage, right? So you have to actually look for these things, whether there's any sex linkage involved, whether it's a dominant allele or recessive allele. In this example, hemophilia it's a sex-linked disease. So the abnormal allele is recessive, right? And the gene is sex-linked. So then the next step would be to work out the genotypes of the parents from the information given and use this to create a genetic diagram, right? So you can be given questions like this, right? So that's about the diseases which are caused due to genetic mutation.